name is Lejane and I chose prompt 1 for this exam. So from class, we learned that Watson and Cribb helped establish and distinguish DNA structures. Base pairing, they established that base pairing between two strands of nucleotides creates a balanced width of helix. Said the helix is comprised of a nucleotide as seen in this picture. From left to right, it contains a negatively charged phosphate group, a 5-carbon sugar. Here we see a deoxyribose sugar since the 2' carbon doesn't contain an oxygen group. If it did, it would be known as a ribose sugar, which is typically seen in RNA, but here we see a DNA. And nitrogen bases here as T, G, C, and A, but had it been a ribose sugar, we would have seen a U replacing the T nitrogen base in this example. Take a closer look at the nitrogen bases. Up top, we see the purine nitrogen bases, adenine and guanine, and they are 2 ring bases. The thymine and cytosine are 1 ring bases known as pyrimidines. The way I remember this is A and G are the first letters for the word angel, and angels have 2 wings, just like the 2 rings of purine, and angels tend to be pure, which sounds like purine. Now that we have unbound the double helix, we can see from the simplified diagram the complementary base pairing between the nitrogen bases. Adenine here in red pairs with thymine seen in green through 2 hydrogen bonds as illustrated by the blue connecting dots. Guanine, on the other hand, pairs with cytosine by 3 hydrogen bonds. The hydrogen bonds are relatively weak, and their main purpose is to provide specificity for the complementary nitrogen bases, which exist in equal proportions to bind to one another. Up top with the orange circles, we see the sugar phosphate backbone that is bound by the phosphodiester bonds. As you can see by the illustrations, the 2 strands run anti-parallel to one another, and that's because the 3' end of one nucleotide binds to the 5' end of the next one. As you can see by the markings on the purple pentagon, each carbon seems to have a purpose. The first carbon attaches to the nitrogen base. The secondary carbon helps us distinguish that's DNA because we don't see a hydroxy group on it or an oxygen. The third binds to the next phosphate, and the 5' carbon binds to that specific phosphate. This creates for us a center that is hydrophobic and scared of water, and a backbone that is hydrophilic and loves water. It is through the hydrophobic interactions that we stabilize the helix. The interior is so scared of water that the nitrogen bases stack on top of each other to prevent water from interacting with them. Different kinds of DNA. However, the most biologically relevant that Watson and Crick unveiled is the one that is demonstrated by the green arrows and demonstrates right-handedness. Right-handedness can be determined by thinking of a spiral staircase. If the rail is to be touched by your right hand, it is right-handed DNA, unlike the ones that are demonstrated by the red arrows. Another important message is major and minor grooves are found on the double helix, and major grooves seen by the bigger arrow are favored by proteins because it allows for surface adhesion. Salt concentration buffer would raise the needed melting temperature. That is because the phosphate group, which is negatively charged, will be neutralized by the positive charge found in the Na plus of the NaCl, as seen by the picture below. Ultimately, this leads to stabilization of the entire polymer because we no longer have the negative charge from the phosphate pushing the strands away from each other. So we need to raise the temperature in order to melt the DNA molecule, and a more comprehensive explanation is found on the slide. That concludes the exam. Thank you.